All right, so we're going to move on to the second part of the review, focusing on the genetic aspect of it. Let me take a look here. What we got. So quizzes 7 and 8 address DNA structure, DNA replication process, and then the purpose of replication is so cells can divide. So then we get into stages of mitosis. And then following that, we finish up with gene expression and how DNA is copied into RNA and how the RNAs work together to build proteins. All right, so this is what we're going to focus on here. Try to make this one a little bit shorter. So what you need to know, a few things. You don't have to draw this, but you should be able to recognize it. And the reason to recognize it is because in drawings, it's going to be simplified and certain parts of it are going to be referred to over and over again. And if you don't know what those terms mean or what we're actually talking about, it's very confusing. So when someone says it attaches to the 3' end, or this happens on the 5' direction, that's kind of meaningless unless you know what is 3' referring to, or what is 5'. And if you don't know what that is, then all that information isn't very helpful or useful. So DNA molecules and RNA molecules are made up of smaller units, like proteins are made up of individual amino acids over and over again, or complex carbohydrates are made out of sugars attached to each other. DNA is made out of actually sugar molecules and other things attached together. Okay? And then they repeat over and over and over again. Those things are called nucleotides. Okay? The nucleotide has a base sugar that's a 5-carbon sugar. So number 1, 2, 3, 4, 5. So there's 5 carbons in this sugar. So every carbon should have an oxygen, and then they should have the hydrogens. However, we see some things that vary here, right? So this carbon here has got an oxygen that's attached to a phosphate functional group. That's part of the nucleotide. Okay? This carbon's oxygen is part of a ring structure. This carbon's oxygen is here, is this OH group. This carbon's oxygen is in DNA missing, right? It's called deoxyribonucleic acid, okay, DNA. Deoxy, without oxygen, it's missing an oxygen. So the sugar is missing an oxygen at the number 2 carbon. That's important. And this one here, the carbon's oxygen is involved in a bond to something called a nitrogenous base, which refer this with an N. And the N is going to represent the things that we usually refer to when talking about DNA and RNA as the A's, the T's, the G's, and the C, not another G, and the C's. Those are the nitrogenous bases. And then in RNA, we'll have U or uracil instead of T. So that's what we have as these nitrogenous. So we get a sugar with a nitrogenous base and then a phosphate. Now, the phosphate's attached to the number 5 carbon. The nitrogenous base is attached to the 1. And then here attached to the number 3 carbon is the OH functional group that's still open, available, and free to form bonds with other nucleotides. So that's how we're going to build longer chains of these, one attached to another, and so forth, is at that location. That's why it's important. So because there are carbons, we're not drawing the structures out here, because there are carbons in this structure, it's like carbon, nitrogen, so they alternate, they're unique for those structures. They have carbons in them, so that's like a number 1 carbon, that's like a number 2 carbon, so it's confusing. The sugar carbons have a little tick mark like this, referred to as a prime. All right, so this one's called 5' phosphate, and this one's called the 3' OH. That's just because it's the number 3 carbon or the number 5 carbon. So the numbers 3', 5', refer to the carbons of that sugar, and that's why I drew the sugar structure here. Because again, those are terms you can't get away from, and you're going to hear. So you may as well know, where do they come from? What do they actually mean? So they're referring to sugar structure. And so we have nucleotides as the core unit of DNA and RNA. So they're polymers. There's groups of them that are joined together. So how are they joined together? Well, what we see is sort of a backbone of sugars and phosphates. So what happens is there's going to be another nucleotide here, sugar, again, simplifying the structure. Instead of a single phosphate, it would be a triphosphate. This, and that could be like that's really almost like ATP, adenosine triphosphate, right? But this with DNA, there's missing an oxygen. In RNA, there'd actually be an OH here, and that would actually be the same, exactly the same as the ATP made in the mitochondria. It's actually it's part of the DNA, as well. And then so are all the others. So the T, same thing. The G, the C, the GTP, they're all pretty much the same basic structure. The energy molecule ones are ribose sugar, and where they have all their oxygens. The DNA ones are missing an oxygen. They're deoxy. So what happens is then a bond forms here between these. It breaks off phosphates for energy, and you have two joined together. And then this happens, again, and again. Phosphates are broken off, a new bond is formed, a new OH end. I'll just put in here like this. And so what's happening is I'm adding ones and making this longer. As I'm doing it in a certain direction. I'm doing it in a 5'. Now, right, this is a new OH end. Every time I did a new OH and a new OH, that's the 3'. So it's a 5' to 3' direction to build. All right, so if we're building chains, like long chains of nucleotides, one after another, after another, after another, there's a certain way in which they're connected, a certain direction in which they're connected. And that's this direction, 5' to 3'. So DNA and RNA are made up of nucleotides. Nucleotides are joined together with bonds called phosphodiester bonds, that are the bonds between the sugars, the phosphates obviously involved. And when they're built in a particular way. Now, DNA is also double-stranded. So now we're going to simplify this even more and put, say, a phosphate up here and an OH down there. Second strand over here. And the way I'm going to do this, you're going to see it's going to be flipped. So I have A, T, C, G, G. I'll do two G's like that. So DNA is double-stranded. The strands are complementary to one another. So what that means is that we have sort of things called base pair rules, which are important for you to know, because they apply to reading the DNA to make RNA and a lot of different processes. So the DNA replication process, the transcription process, all involve base pair rules. All right. So if you don't know them, you're lost in a lot of different things. Okay. So base pair rules are that in one strand, an A will pair up with something on the other strand. It'll either pair up with another A, a T, a G, or a C. It has a bunch of options. A's pair with T's. And then G's pair with, well, all we're missing is the other one, right? Is the C, like that. So A's with T's, G's with C. So that means if this strand says A, T, C, G, G, that means we know the code, another color, of the other strand. It's going to be T, I'll explain why I'm doing this upside down in a second, A, G, C, C, like that. So A with T, T with A, C with G, G with C. Okay, so that's complementary base pair. What happens? So how do they join together? What do they do? There are hydrogen bonds between them. So the A's and the T's will hydrogen bond with one another. G's and C's will hydrogen bond with one another. A's and T's have 2 bonds. G's and C's actually have 3 bonds with each other. And that's how the 2 strands are held together. Now, why did I draw this one upside down? The strands are anti-parallel. So it's not phosphate and phosphate. So you can see they're kind of opposite each other. One is upside down compared to the other. That's also important in terms of the DNA replication process and why it works the way it works. So it's important to know that part of the structure. Structure and function. Process is going to be dictated by and affected by the structures of the molecules involved in the process. And so if you understand the structures, it'll help you understand the process. Okay. If you don't understand the structure, very difficult to understand the process. You'll just be kind of lost or always wondering, why is this? Or it doesn't make any sense to me. But if you know the structures, say, oh, that makes a little bit more sense, at least why it's different or why it doesn't happen like this or why it does happen in a particular way. So keep in mind, it's very important for you to understand the structures as much as you possibly can. All right, so structure of a nucleotide, how nucleotides are joined together, how strands are joined together with base pair rules and hydrogen bonds. Again, there's way more detail to it, but again, broad review, big picture, this is what we have. Now, DNA stores information. That's the last part we'll get into, is how the information is used. So DNA doesn't really do anything. Okay? It has no action or activity that it carries out in a cell. It doesn't do anything at all. Right? RNA carries out activities. Proteins carry out cellular activities. DNA holds information. So DNA is like an instruction manual or a textbook. You're not going to learn anything from a textbook if it just sits there. You can carry it, you can put it on your head, but nothing will happen to it. Same thing. You just stare at something and nothing's going to occur, right, unless you actually do something. So say when you're studying, drawing these structures helps you learn or remember them. So carrying out some kind of activity is what's important. However, what we need to look at is, first we're going to look at how does the DNA replicate so cells can divide as a process, and then we'll come back to looking at how DNA stores information. So the first thing is, we started the class, cell theory. All cells come from other cells. So cells will divide through a process of cell division. Bacteria use something called binary fission, and in eukaryotic cells, it's mitosis and cytokinesis, or cell division processes. So the mitosis is kind of divided into karyokinesis and cytokinesis, but we're not going to get into all that. So essentially, there's division of the genetic material and then division of the cell itself. We're going to kind of combine that into mitosis. Well, in mitosis, this cell and the cell that we started with, and then the 2 new cells we end up with should be exactly the same. If we have DNA in this cell, that means we have to have DNA in this cell. And that means we have to have DNA in this cell, and that means the DNA in this cell and this cell have to be the same, not half the amount of DNA. And if these cells were to divide into more cells, same thing, they'll all have to be the same as the original cell. So how is that going to happen? Well, it means before we divide, we have to double the DNA. Double DNA before cell division. So we get into the process of DNA replication. And so there's simplified ways of going through it, and then there's the real ways that it actually occurs that are a lot more detailed. And we're going to do something that's a little bit in between those two. So from the most simplistic to add a little bit more of the real process to it, but not everything. So we have DNA, phosphate, OH, phosphate. Let's say we have our A, T, T, C, C, A, G, A, C. I'm just put a little short piece. That'd be T, A, G, G, T, C, T, G. Okay. The strands are going to get pulled apart. Now, this is not exactly, this isn't what really happens, but this is the generic sort of explanation of the concept of how it happens, not the details of what really occurs. Okay, so what really occurs is more complicated than this. But essentially, the two strands get pulled apart. So an enzyme can pull apart these two strands, an enzyme called helicase. All right, and then we got the two strands like this. I'm not going to draw this. Okay. And then what will happen is that an enzyme, a DNA polymerase, will make new strands along these existing strands. And I'm actually going to, just to make this actually easier to even look at, I'm going to flip this one. Again, because I said I'm not doing this exactly the way it really occurs, but this is just more the concept first. Okay. Okay. So what the DNA polymerase is going to do is it's going to read a strand of DNA in a 3' to 5' direction. So in a OH to phosphate direction, it'll read it and it'll make new DNA 5' to 3', like this, in this direction. So that means it'll kind of say, oh, there's a C here. What do I need? G goes with C. There's an A here. What do I need? A T. A T will go here. What will go here? A G, a T, a G. Okay, actually, I'll leave that because I'll talk about something else a little bit later. When mistakes occur, what happens? A, T, T, so on. You can see this sequence of this one here, I did in green, T, A, G. Now there's a mistake there. That's on purpose. And new double-stranded DNA. So essentially what we have is something called semi-conservative replication. Semi-conservative replication is where we keep one original strand completely intact and then build a new strand along it. We use that original one as a template. The same thing's going to happen here. I'm going to build in a particular direction. I'm going to go, oh, I did this wrong. Yeah, that should have been the OH, because they were opposite each other. So now, again, the same thing is going to happen. I can build it 5' to 3', but now I have to build it sort of the opposite way here. OH like that. So that would be our A, T, T, C, C, like that. And that should line up with the first code along the top here. Okay, they'll hydrogen bond, with each other and so forth. So now we'd have two new double-stranded DNAs. It'd be exactly the same as the original, and then eventually, through the process of mitosis, they could be separated into separate cells, and then each cell has the exact same material as the original. That's, again, a great oversimplification. Does not really happen like that, but many of the things I said are true and are rules that happen. It's just that there's way more to it. There's a lot more complicated things going on than this, and I'm leaving out a ton of other details. But what is important right now? Base pair rules, okay? Directionality, okay? That the new strands have to be built in a certain direction. The template strands have to be read in a particular direction. That part's going to be important, okay? So now I'm going to get at a little bit more of the detailed part of it. And add a few things into it. Again, not everything, because it becomes very complicated, but I'm going to add a little more to it. Now, what's often left out And even in majors biology courses and textbooks and explanations, and how this process works is that it's going to happen along both of the original strands at the exact same time. But because those strands are opposite one another, we usually draw pictures like this where they seem to be independent from one another, but they're not. The enzymes doing the process are connected to one another. So they could only be working in one direction. That means one of those strands are going to be read correctly, whilst the other one is opposite, so it's going to be backwards. Yet everything still has to happen following the same rules. So let's see if I can explain this here to you. Okay, DNA pulled apart. Okay, so maybe here's a phosphate, here's an OH, get ends go to that. Now, we would have our ATT. And I'm just randomly making these things up. You don't have to worry about actual sequences of things that I'm doing here. They're just totally made up every time I would do them. I'm probably doing them different. They're not specific genes or anything like that. Okay, so they're pulled apart by an enzyme. Remember I said an enzyme called helicase pulls the strands apart. The DNA enzyme, the DNA polymerase, can't just make stuff from scratch. It requires an RNA primer. So it actually needs a little bit of RNA to be attached to these strands here to build off of. And then what's going to happen is then that enzyme can come in, the DNA polymerase, and it can build the new DNA, just like I was talking about and showing you here, from that primer, and it can now start to make all these new pieces, the G, T, C, G, and so forth, as long as it's going in the correct direction. So what's going to happen, though, is one of these strands is going to be read 3' to 5'. So that'll be making the new DNA opposite that, right? 5 to 3, continuously, so non-stop. We'll call that the leading strand. And it's continuous. Replication. The other one, has to follow the same rules. So it could only make DNA in a 5' to 3' direction from a primer. But that means the opposite direction. So that means there has to be an enzyme down here. Many books and things are going to show these disconnected, but they're actually attached to each other. There's other proteins in between them, but they're physically connected. So, it's like, so how do they both connect to one another? How do they both move in one direction along this opened up DNA and then both follow all the same rules? Well, for one of them, it's easy. It's just moving in the correct direction. And that's why we call it the leading strand. The other one, it's hard because it's backwards. Right? So this one is the one called the lagging strand. And what happens along the lagging strand is what's referred to as discontinuous replication. So discontinuous replication means start, stop. So that means it's going to start and make a little piece of DNA, but then it just stops. And then what has to happen is along this strand, another piece of RNA, a primer, is made. And then this enzyme will have to leap forward, grab onto it, make another piece of DNA here, but then it will stop. There'll be another RNA primer. And then as this one's just working continuously, non-stop, continually going, this one has to keep jumping and then making pieces, piece, like that. That's what we call the lagging strand, discontinuous replication. There's more detail in the lecture. There's stuff in your book. There's a couple of video links and things to animations to show the process. But what you need to take away are some of the terms, really specifically, of the process. So when DNA replication occurs, you will, in the end up with 2 new double-stranded DNAs that should be identical to one another. for one of the two strands being used as a template, once the process begins, it's nonstop. The enzyme who does this will read information and build something that requires energy. So, right, so where's the energy come from? Remember, what are these things? These things are sugars with nitrogenous bases, like adenine or guanine attached to it. And then what are they? Well, they exist in triphosphate form. So they exist as ATP, GTP, CTP, and TTP. They A, G, C, T. This is the genetic information, but the individual units are these energy molecules, these triphosphate molecules. So they provide their own energy. When they come in, they're holding on to these extra phosphates until they're attached. And then these things get broken off, and the energy is used to make the new strands and actually build them. So they provide their own energy to be built. So the enzymes can build these structures. For the lagging strand, we said it's much more complicated because we have discontinuous start-stop replication with many primers being required, and the enzyme being kind of pulled in one direction but wants to work in the other direction. So a lot of other stuff goes on, which is kind of beyond what you would need to know from non-majors sort of class, except that it happens and the rules why it happens. But you can look up. I give lectures on the details of it separately, but you can look this up if you want to see all that or have the explanation of exactly how it happens. One of the last things I want to add in here, though, is that there's RNA, and then in one of my previous examples, I made a mistake on purpose. I showed like A with an A or a G with a G, and not the right nucleotide. So what happens here? There is ultimately an enzyme that comes in here to do what's called proofreading. And a proofreading enzyme will check, is A with A, or I'm sorry, is A with T, and if it is, okay. And then if that is correct, it moves on. Is T with A? Is that correct? It moves on. If it finds mistakes, it will correct them. So the first mistakes that it will find are that some of these pieces are not even DNA. They're RNA. So the proofreading enzyme will actually cut away the primers, and it will then replace them with the appropriate DNA. And there's more to this about joining them and a ligase enzyme and other stuff happening, but that's kind of the basic idea. It has to work a little bit on the leading strand, a lot on the lagging strand, replacing lots of primers. If it finds mistakes, say a C with a C, it will then cut out the mistake and then go ahead and replace it with the proper nucleotide. So that should happen. If it doesn't happen, that leads to mutation, which again will be in the beginning of the third exam material. Thus will connect as well, because we'll talk about genetic diversity within organisms, and one of the ways that they become different is through mutation, and one of the types of mutations is just natural mutation through DNA replication, where errors are not fixed. So the two DNAs in these cells may not be identical. One of them may have a few differences in the sequence that will lead on and be carried on and carried on and carried on. It could be good, it could be bad, it could be neutral. Don't know what it'll be. It's random, how it happens, how the mutation occurs. So there's that. Okay. So we have structure of DNA, the overall process of DNA replication, and now the purpose of DNA replication was for division. So again, there's a whole worksheet where you're actually identifying stages of mitosis, you're counting stages of mitosis from a variety of different photographs, and then calculating how many are in each of the stages. So definitely work on that worksheet, because in addition to being one of the worksheets you have to do for the class activities, it's studies. You're studying for this part of the exam. So you need to do that now, like before the exam, if you haven't already done it. What it will go over is how a cell will have its wow, that's really terrible. It's double-stranded DNA. Okay, there you go, like that. DNA replication, we often show them being separated, but that's not really what happens, okay? The one double-stranded DNA is essentially joined to the other double-stranded DNA. There are proteins that actually connect the two after DNA replication. They're not separate. They're actually held together, right? And they're called sister chromatids. And then the process of mitosis will eventually pull those apart. Cells usually have more than one chromosome. They have many chromosomes. So this whole process has to be coordinated with all the chromosomes of the cell all at the same time, so they can all be separated properly into two different cells. And that's the process, again, of mitosis. So what will happen here is that we describe these things as humans in how we can actually see them. The process does not occur so that we can see it. The process occurs so that it functions properly and accomplishes the goal that it needs to accomplish. We can see certain things at certain times with our eyes through a microscope, but they're not happening for us. Again, and the DNA isn't doing anything. The chromosomes are inanimate. They can't move on their own. So when people say chromosomes line up in the middle of the cell, they're not fish. They can't move. They can't line up in the middle of anything, right? They're not. The DNA isn't alive, and it has no machinery mechanism to move. Proteins will move the DNA. Proteins will attach to the DNA. Proteins will pull on the DNA, and they will try to separate. So it's the action of proteins that are going to cause all the things that we see. So what do we actually see? We'll see DNA condense. It doesn't condense itself. Proteins condense the DNA, meaning they make it really tight into like a little package, as opposed to just like loose string or yarn or electrical cord. It's coiled up and rolled up into a nice little tight ball. Okay, so it's organized, and that's going to happen in this process. And then we'll be able to see it, right? But that's just a aberration that we happen to be able to see it at that stage. But that's how we define these things. So in any case, what happens is DNA replicates first. Then when it gets prepared to divide, it does condense so that it's more organized. So it's not all stringy and tangled when it tries to pull apart. Okay. We call that prophase, where we can usually see chromosomes, in a cell. Again, they're not doing it for us, but we can actually start to see chromosomes in a cell like this. Now, we don't see them everywhere in the cell. Usually we see them held tightly inside a nucleus, okay? And that's prophase. Eventually what will happen is that the nuclear envelope will break down. The nucleus, the membrane, breaks away, and those chromosomes are kind of spread out then anywhere they want to go in the cell. But the other thing that's happening is that in the cell are structures that build microtubules. They're called microtubule organizing centers, MTOC. And they'll shoot out like a stringy filament, and they'll attach one to each of these pairs like that. They couldn't do that in prophase because they're held inside a nucleus and held inside the membrane, so they can't attach anything to them. It's during the next phase called prometaphase when this happens. And both of them will do it. They both shoot out these little stringy filaments. They're called kinetochore tubules. Okay, so if you want to know what the detail of them are, that's what they're called. Now, once they attach, like a fisherman, they're going to try to reel it in. Then this mechanism actually shortens the proteins, actually cuts off units and starts pulling, and these little strings, these microtubules, shorten, and they actually get. And what will happen is that as the tubules shorten and pull on the chromosomes, what we'll find is that, the pull from this side isn't any stronger than the pull from that side. They're equal to one another. So it's a tug of war. This one's pulling this direction. This one's pulling in the other direction. Neither one is stronger. the sister chromatids are held together by a little ring, cohesin protein that's holding the two together, and that they can't be pulled apart. It's like a little belt like wrapped around the two of them and just cinching them tight together, and they cannot separate. So what happens is just a tie, a tug of war that ends in a tie called metaphase. And they are essentially lined up in the middle of the cell. Now, eventually a cellular signal binds to this protein, this cohesin protein, and removes it. So it like opens the belt up. Right? And now the two sister chromatids dropping this off, can be pulled to opposite sides of the cell. So there's action here, there's motion, there's movement. When you see photographs of these things, they often don't, they might look closer, they might look further apart. They're all different ways because this is a stage of motion, this anaphase. And then eventually, when they get to opposite sides of the cell, the DNA will start to decondense, meaning you can't quite see individual chromosomes anymore. And then new nuclear membrane starts forming around them. And then the cell itself will actually start to have the membranes pull inward through a cytokinesis process and actually divide into two new cells. And that would be telophase. So again, you should make sure you identify actual stages of real cells in the assignment that you're given, where you're going to find them, identify them, and then count them, and then calculate how many are in each of these stages in different groups of cells. I think you have about 30 or 50 cells to count in different photographs. So you should get a lot of practice identifying these stages if you do that assignment. So make sure, again, you do that, and it should all come together for you. And this part should be pretty easy. Lastly, the DNA doesn't exist just so that it can be replicated. So yes, it needs to be replicated or cells can't divide into more cells. But, why do we even have it in the first place? What's its job or role? Well, the DNA provides information for RNA. A, T, G, G. Okay, so here's some DNA. What's going to happen is, in a process called transcription, not both, but one of these pieces of information are going to be copied. So another enzyme will come in, an RNA polymerase, and it will just read one of the strands, not the other, and it'll make now instead, the way you know it's different is instead of T, we have U, right? Like this. You can tell that this piece up here in pink or red is RNA because it has U's instead of T's. But otherwise, it's the same sequence. You look, A, G, 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 A, we see, oops, yeah, that should have been a C there. Okay, G, and so on. So, okay, so it should be the same as this strand here. So this is the one with the code, but this is the one that's used as a template. And then from that, we get this piece of RNA. And that's the process of transcription. To transcribe means to copy. Right? So we're copying a piece of DNA into a piece of RNA. Now, the significance of that is DNA stays in the nucleus. It's kind of like an important book that stays locked away in a library, can't be loaned out. Maybe people can't even touch it. You know, it's special information that should not be messed with, right? But if there's information that's valuable that people need to access, that they want to use, they need to be able to look at it. They need to be able maybe to take a copy of it, maybe Xerox it away, and then do something with it. Maybe use the information. So that's what RNA actually uses the information from the DNA to do something. But first, it has to be copied, and so the copying part is called transcription. So it's very similar to DNA replication, except a lot more simplistic, because only one strand is copied, not two. And then this will leave the nucleus. So DNA is in the nucleus. The RNA will leave the nucleus, goes to cytoplasm. And that's where it'll work and carry out its job. Now, when RNA is made, it could be one of several different types of RNA. So one type of RNA is going to contain a code for a sequence of amino acids. So that means that the A, U, U, G, G, A, C, C. Oops, yeah. So that's the RNA, the code here. That's going to be used and eventually translated into another language, okay, a language of amino acids from a language of nucleotides. That's going to be one type, the type that we call messenger RNA. There's another type of RNA that's going to do the decoding. It's one that's going to actually then say, okay, now I'm going to see how this works. So let's see, U, A. Now the whole thing's made up of A's and U's and G's and C's and C's and U's, but it's folded, okay, into loops and stems. This is called transfer RNA. Again, you should see the details in the real detailed lecture. The transfer RNA carries with it an amino acid. And that amino acid is very specific, and it's related to whatever the code is down here. And that code's called an anticodon. So it's the very bottom of a transfer RNA molecule. There are 3 nucleotides that are an anticodon. They would pair up with a messenger RNA codon, which is also a group of 3. So see, base pair rules, A with U, U with A, U with A. So these 2 things would actually match up like with one another. And what that means is that this code, A, U, U, would code for whatever this amino acid is. And this transfer RNA would be the go-between, would be the way that it's being translated. Yet there still has to be some kind of mechanism or machinery that can actually put all the things together. And that's where we get other very complicated, which I'm not going to really bother drawing, like pieces of RNA called ribosomal RNA. And there are 2 parts. There's the large subunit and then the small subunit. The large subunit has all these like binding sites, places where certain things can attach to it. And then collectively together, the 3 types, the messenger, the ribosomal, and the transfer RNA will work together to carry out the process called gene expression. That's what we're going to finish up here with this, is to look at that and how they interact. So we have DNA holding information. The information is copied from the DNA into RNA. The RNA is already determined to be one of these types. It has to be processed. So you have to change it slightly to make messenger RNA. You'll have to change it. You have to fold it up to be this transfer RNA, and then something has to attach amino acids to it. That's a whole separate process on its own. Then you have these ribosomes. So you have different pieces of ribosomal RNA. They will have to all fold. They'll join together, and they'll also join with proteins, and then collectively they'll build these structures called ribosomes. All right, the large ribosomal subunit has 3 binding sites. Okay, it has an E site, P site, and an A site, like this. We have messenger RNA, which stays like straight and linear, right, like that. Okay, and that just is what's going to keep displaying our sequence or code of nucleotides like A, U, G, C, C. And so on. So you get this long code. All pieces of messenger RNA code for different genes. So a gene codes for a piece of information, but that piece of information is encoded in the messenger RNA. The messenger RNA then has that information organized just into a sequence of nucleotides like this, but then it's going to get further organized into groups of 3, the codons. And each codon codes for 1 amino acid. So what will happen is a transfer RNA who will carry the first one, the very first amino acid, which is always going to be methionine. It'll come in, well, it'll do a whole thing. It binds to the 5' cap, it reads, it finds a start codon. This is the one it pairs with, AUG, that's the start. And then the process will begin. Right? And so what will happen is you'll get the transfer RNA into the A site. Oh, sorry, that's what normally happens. It normally goes into the A site. But in this particular case, for the very, very first one, the very first time, methionine actually goes into this middle site. So it's unique. After it does that, then another one will come into the A site. So it'd be like amino acid number 2. This ribosome will be checking to see if these nucleotides in the anticodon match up with nucleotides in the codon. And if they do, then it will create peptide bonds between amino acids and join them together. When that happens, this whole structure will essentially shift so that now this is the E site, and this is the P site, and this is the A site. So what you'll have is in the P site, you'll have a transfer RNA with a peptide, a chain of amino acids. The E site will be empty transfer RNA. There's no amino acid attached to it. And the A site is available and open for a new transfer RNA to come in. So like amino acid number 3 comes in, it'll read this anticodon. If it matches up, then it says, oh yes, and then the large subunit breaks the bonds here, makes a new bond there. This guy gets kicked out. The empty transfer RNA gets kicked out, and the whole thing just kind of shifts, and it just reads. So this is the process of translation, also referred to as protein synthesis. This is the process where all the different types of RNA work together as a team. Transfer RNA, messenger RNA, ribosomal RNA. The messenger RNA carries the code, the information. The transfer RNA is a way of decoding it from nucleotides to amino acids. And the ribosome is the machine that puts it all together. It kind of puts all the pieces together, reads them, makes the bonds, and then keeps moving along until it starts to build then a longer chain, a polypeptide, which will ultimately, through a whole bunch of processing, become hopefully functional and become a protein in the cell and actually do some kind of work. So that's again simplified. There's a lot more to it than that. There's places, where this occurs. Normally it's occurring in the cytoplasm. It can associate with the endoplasmic reticulum. These ribosomes can attach to the membrane of the ER, and then it could force these peptides inside it. And that's where they'll get folded up and things will get added to them or removed from them so they can become processed to become functional proteins that will go to a future destination, either in the cell or outside of the cell. Or they might even go back, they might go into the nucleus. It might be one of the proteins that work in DNA replication. They have to be made outside in the cytoplasm, and then they'll eventually come back in. So just make sure you kind of have all the terms that are on the sheet. And then there's a few of the questions that you should be able to answer kind of related to these sketches. And if you can do that, you should be in pretty good shape. The inheritance part, the Mendel part and all that stuff, and meiosis is then on exam 3.